Transposable elements in eukaryotes. The existence of transposable elements was first detected in Barbara McClintock's studies of maize in 1940s. She was awarded the Nobel Prize in 1993. These elements are fully comparable to those in bacteria and were detected by their effects on the expression of certain maize genes. Maize contains several types of transposable elements. These elements have a sequence of organization similar to their bacterial counterparts. They have inverted terminal repeats, encoded transposase, and generate short directed repeats in their target when they transpose. They are called autonomous because they have the ability to transpose. They are members of the same transpose family, are often present in maize, but are unable to transpose on their own because of internal deletions have destroyed their transposase gene. These copies are called non-autonomous and are incapable of movement unless transposase is supplied by another transposable element. Non-autonomous elements retain the inverted terminal repeats at which transposase acts. The best characterized maize TN element is 4.5 kb element called AC for activator. Defective versions of AC are called DS for dissociation. As in bacteria, transposons in maize are associated with a variety of structural aberrations of chromosomes. Deletions, duplications, and inversions may occur at sites where autonomous elements are present. The transposition of maize elements is regulated in part by the development of the plant. Unknown signals generated at certain periods of plant development affect the frequency of movement of these elements. It is possible for an autonomous element to become reversibly inactivated, that is, incapable of transposition. Presumably, such a change results from secondary modification to the DNA sequence of the element. Cycles of methylation and demethylation of certain DNA sequences may be involved in this process. So, transposons have been detected in the genomes of other eukaryotes, including those of yeast, Drosophila melanogaster, and humans. They are probably present in all genomes. Researchers are exploring them in model organisms for basic studies on the regulation of gene expression. The so-called P element in Drosophila represent a long about 3 kb sequence with inverted repeats at its ends. Modified versions that lack the transpose enzyme activity have been constructed in the laboratory. This once inserted in fly's genome, it remains in a fixed position and can be used to generate frame shift mutations or to serve as a genetic marker. As well, the P element can be engineered in the laboratory to contain a foreign gene. When inserted into a bacterial plasmid and subsequently injected into a fruit fly egg, the P element is integrated into the embryo's chromosome, creating transgenic animal. Like the transposons described for bacteria, eukaryotic transposons cause unstable mutations in genes that are reverted to wild type when the element is deleted. In addition, these transposons cause short direct at the site of their insertion. However, some of these elements have a fundamentally different genetic organization and mechanism of transposition than the bacterial elements. Retroviruses infect eukaryotic cells and insert in host genome. These eukaryotic transposons are similar in organization to the proviral form of retrovirus, which are eukaryotic viruses that contain a RNA genome. The most famous example is the human immunodeficiency virus or HIV, which causes AIDS, acquired immune deficiency syndrome. Retrovirus introduce an RNA molecule into cells that can be converted into double-stranded DNA by a unique viral encoded enzyme called reverse transcriptase. The double-stranded form of virus can integrate randomly into the genome of the infected cell, where it resides as a provirus. After integration, the provirus is replicated as part of host genome. The proviral form can express viral genes as mRNAs. which then translated to host into viral proteins that make up the capsid. Capsid proteins are the mRNA itself that constitute the viral genome assembled into infectious viruses that are then shed from the cell. The integration of the retroviral provirus into the host genome generates short direct repeats flanking the viral sequence, like those seen in bacterial transposition. The retroviral infection cycle, therefore, has features in common with transposons. Imagine a situation in which a genome has occurred a number of retroviral proviruses integrated at various points. The proviral genes continue to be transcribed by the host RNA polymerase. Over time, mutations may occur that inactivate or alter the proviral DNA sequence so that functional viral proteins can no longer be produced. The proviral element could then be regarded as functionally defective. However, as long as functional reverse transcriptase like activity is produced by the element or its relatives, the mRNA can be converted into double stranded DNA. It then will be able to integrate into new genomic locations by a process resembling bacterial transposition. The important feature of this pathway are that the appearance of the proviral DNA form at new genomic locations amounts to a transposition event, and that the transposition process involves an RNA intermediate. This relocated gene is called retrotransposon. Retrotransposons make up a large proportion of human genome. Retrotransposons in the human genome are believed to represent vestiges of retroviral infections that occurred during an earlier era in human genome evolution. Presumably, as a result of mutation, most of the original retroviral DNA sequences eventually lost their ability to transpose and hence now remain in a relatively fixed position. A relatively small percentage of them have, however, retained the ability to relocate. As mentioned earlier, such retrotransposons occupy a huge proportion, approximately 40% of total human genome's DNA nucleotide sequence. Such transposons comprise the moderately repetitive DNA described. There is no evidence that they code for proteins, and hence they merely occupy, in a physical sense, a major portion of the human genome. Some examples do, however, bear a close relationship and resemblance to legitimate genes, but since they do not generate protein products, they are referred to as pseudogenes. They have they Why they have persisted during the evolution of the human genome sequence is unclear. As a first glance, it might appear that retrotransposons merely represent parasitic DNA that has not yet been eliminated from the genome. Conversely, it may be the case that during the phylogenetic history of human species, transposons plays a key role in creating new genes and or regulatory sequences. So enhancers, silencers, etc., and have not yet been detected. Indeed, several functional genes that have sequence resembling ancient transposons have been regulated in the human genome. These include telomerase, an RNA containing enzyme that is responsible for the synthesis of so-called telomers, chromosome ends, and splicing genes involved in gene rearrangements of the human immune system. Yet, one additional consideration is often put forth. Perhaps retrotransposons now function to facilitate the restructuring of eukaryotic chromatin, including folding and looping, which characterize transition in from that occurring during the DNA replication and gene expression phases of chromosomes. Similar considerations concerning persistence of seemingly non-functional DNA is also to introns. As human genome project data is interpreted, a more complete understanding will be achieved of what, if any, functional significance retrotransposons and introns possess. And the sequence or arrangement of nucleotides in genes is completely evolving, either by random mutation, by occasional recombination events, or from the action of transposable elements.